Hello, my name is Sandra and I will be presenting this paper called Astrocytes and Glutamate Homeostasis in Alzheimer's Disease, a disease in glutamate synthase but not glutamate transporter 1 in the prefrontal cortex. This paper reflects research done by a group of researchers from the Czech Republic. For some background, we all know that Alzheimer's disease is a progressive neurodegenerative disorder, and it's characterized by both behavioral and molecular phenotypes. On the behavioral side, we see loss of cognitive function, personality changes, mood alterations, and memory deficiencies. And molecularly, we see loss of neuronal connections, neuronal decay, and most iconically, these extracellular beta amyloid plaques or built up. Now, astrocytes are glial cells that protect against exotoxicity, and they are the key element in maintaining homeostasis in the central nervous system. They're important to us because they also contain glutamate synthase protein, and they regulate the glutamate transporter 1 protein, which are both believed to be targets in AD. Lastly, glutamate is the primary excitatory neurotransmitter in the mammalian central nervous system, and it's important for cognition, memory, and learning, 3 areas that we see affected by Alzheimer's disease. This paper explores this one research question, and is, does the expression of glutamate synthase and glutamate transporter 1 protein decrease in astrocytes in the medial prefrontal cortex during the progression of AD? So, in short, to simplify it, it is looking if Alzheimer's disease is caused by a homeostatic imbalance or can be directly linked to a homeostatic imbalance in GS and GLT1 proteins. They hypothesized that the changes in GS expression would influence glutamate homeostasis, and therefore that would lead to a failure in synaptic connectivity, affecting brain functions such as mood, cognition, and memory, all which we see impaired in AD patients. To test their hypothesis, they utilized triple transgenic mouse model, a very known mouse model in Alzheimer's disease, and they compared those mice brains to controls at 4 different age groups to look at the progression of the disease. We had 1 month, 6 months, 9 months, and 12 months. These mice were anesthetized with sodium phenobarbital, and brain slices were looked at under a microscope using immunohistochemistry. As we can see in the control, that astrocyte, that single neuronal cell right there, in the control group, we see a much higher branching amount right here, and then we also see it in the red. We see all these neuronal connections in comparison to the AD mouse brain, where the astrocyte itself is a little oddly shaped and a lot less in connective strength, mimicking an Alzheimer's disease brain. Additionally, they tested for the protein levels of GS and GLT1. Here we see the protein levels of glutamate synthase, and they show a significant decrease in the Alzheimer's disease mouse model at month 6, 9, and 12. However, when they came to look at the glutamate transport protein, they saw a decrease in the transgenic mouse model in comparison to the control. However, that decrease was not significant enough for them to draw any direct conclusions linking the transporter protein decrease to Alzheimer's disease. So with these findings, this was the results that they came to conclusion to, that the glutamate transport 1 expression is slightly reduced. However, it's not significant, so therefore they cannot directly correlate a reduction in the transport protein in Alzheimer's disease. So they believe that glutamate reuptake is preserved in AD. Additionally, they saw that GS expression is significantly reduced. They believe that is linked to disturbances in the glutamate cycle, and they saw that well before these beta amyloid plaques form. And lastly, they saw a reduction in astrocyte branching and size. So what does that mean for us? Ideally, this means that with this high sensitivity of GS to oxidation, it presents the idea of an antioxidant treatment for Alzheimer's disease patients. It also points at astrocytes as a major target. So knowing that they are metabolic stability, they allude to being very therapy targets in treatment and prevention of AD. Thank you so much for your time.